We are surrounded by microbes. We live among microbes. We have microbes inside our bodies. There are 100 trillion of them in our intestines, yet we're not doing so badly. So there's some kind of deal that has been made somewhere. So the question is, when do microbes cross the line? The idea of going beyond that barrier, that's where science really begins in the end. Junk food, in quotes, to simplify things a bit, is a destabilizing factor for the gut microbiota. It brings in excess elements that humans have never been exposed to until now.
Hello everyone, welcome to this episode. Today we'll discuss microbes and their link to our health. Hello, Philippe Sansonetti.
Hello, Christophe.
I'm very happy to be able to welcome you today. So, we're going to be able to talk about a topic that we haven't really covered much on the channel, which is to talk about microbes, bacteria, and to take a look at what's hidden behind the living world. For those unfamiliar with you, can you briefly introduce yourself and share your background?
Hello, yes, of course. Briefly, I'm a trained physician. I quickly became interested I specialize in internal medicine, but very quickly in infectious diseases. I could tell you why, by the way, but really that has been my field and my passion. But very quickly in medicine, I realized that everything was a little too much by the book, as they say in English, and that there was a bit of a lack of seeking knowledge for oneself and not just from books. And so quite early in my scientific and medical career, in the middle of my residency, I took a turn toward research. I managed to get into the Pasteur Institute at the time. It wasn't easy. It was Jacques Monod who brought me in because he saw that I had a bit of a tendency like that to do science. And for several years, I did both medicine and science, hospital medicine and science, which was really fascinating. So it was a kind of feeling of intellectual fulfillment, but at some point you have to make choices. And around the early 2000s, I really stopped practicing medicine. I focused solely on the scientific activities in my laboratory. I went to a laboratory at the Pasteur Institute that was starting to grow. Well, attending meetings abroad made it extremely difficult to be reasonable and serious towards the patients. So it was better for me to focus only on science. I always remained, and I am still very passionate about the interface actually between medicine and science. So the main part of my career then was really running a laboratory at the Pasteur Institute. Then, starting in 2007, I was elected as a professor at the Collège de France. And that was also a great moment because it was a time of sharing, meaning I began to truly share my knowledge with the audience at the Collège de France. And also to share with other professors who were in different disciplines, including those in literature. It was an important moment in my development, if you will, in relation to the field. That's when I also began to understand that you have to explain your discipline and not just keep your head down, only thinking about what you'll do the next day in the lab or with the postdoc or the grant application you're going to write. There were other dimensions to explore, and that the advantage of teaching helped broaden my perspective a little and to become interested, including in aspects at the interface between biomedical sciences and anthropology, for example, or sociology. And that's when I started writing, let's say, for the general public, because I thought there was indeed a need for science communication in areas that are sometimes a bit sensitive. I started with vaccines.
I made a lot of friends and that's it.
That was kind of my journey.
All right. So you said that you stopped practicing medicine to do more research. Now, the difference isn't necessarily clear to everyone. Is that the difference between being a researcher and being a doctor?
Yes, that's true. It's a very relevant question. You can actually do both, and it's even fascinating. Medicine is still about applying medical knowledge to patient care. That is, helping people get better by identifying the source of their discomfort or illness and providing a solution. But by doing that, we realize that there isn't always a solution. And basically, we quickly get the idea to go beyond that barrier. And that's really where science begins. That's where research begins, at least scientific research. It's about identifying medical issues for which there isn't yet a solution, not a truly effective solution for the patient, and trying to move things forward in order to actually offer solutions. In my field, for example, infectious diseases, the issue was, or still is actually, that basically finding new antibiotics is great and it's essential for treating acute situations. Finding vaccines is also good because it prevents the disease instead of just treating it, which is more comfortable for everyone and moreover is much more economical. The economics of prevention is something that isn't really ingrained in the minds of our fellow citizens, but it's essential. So there isn't really a barrier. There's a lot of overlap between, let's say in this specific case, medicine and biomedical research. But there comes a point where it does become two different professions, and it can even feel a bit schizophrenic. When you find yourself in a hospital environment where there's all the tension, the difficulty, including administrative problems and so on, and then you can try all that in the afternoon to switch your brain and move on to the subjects, to the problems in the lab, which are not insignificant either.
Yes. As you said, when we do science, we turn to what we don't know yet, what is still left to discover, and among all the things that are still left to discover and understand, you chose to work on bacteria and infectious diseases. What led you to make this choice?
There was a medical aspect and a scientific aspect. We're coming back to it. It's kind of the two. I wouldn't want to call it a duality because for me it's fine, they go together, but the medical aspect came very early in my hospital career. As an extern, as an intern, I had been very struck by. We're talking about a few years ago. I started my medical studies in 1968. There were already organ transplants, especially kidney transplants. And I was very struck by what I saw in the departments I rotated through, by the fact that basically there was an enormous amount of work from the medical team, the nurses, all the surgeons, everyone involved in the event, like a kidney transplant, for example, not to mention the patient, and that this incredible work could be destroyed in a few hours or in a few days by an infection because the patient was immunosuppressed by the treatments necessary to prevent rejection and became very sensitive to infections, where people who were carrying a tubercle bacillus, for example, would develop a very severe tuberculosis. So that really shocked me, and honestly, that waste, to put it a bit bluntly, but that's really what it was, a tremendous effort, the patient's hope ruined by that, by an infection. So that's really what encouraged me, and also a certain knowledge. When you work in infectious diseases, you're also interested in tropical diseases. And the realization or understanding of health issues, especially for children, because when you work in infectious diseases, you deal a lot with infected children in the countries of the global south. So it was a bit of this mix that, in a way, led me to choose in particular the type of microbe I worked on. Now, the scientific decision, I don't think I was alone in that situation. It was the fact that I found myself at a time which was the era of the emergence of molecular genetics. And when I joined the Pasteur Institute, it was a few years after the Nobel Prize of Loaf, Monod, and Jacob. It's a major Nobel Prize. It was really the discovery through microbes, through Escherichia coli, of gene regulation, which led to the discovery of RNA messenger, and ultimately to realize the fact that we could then do genetic engineering, clone genes, make them express themselves, and do everything we can do today with what we call genetic engineering. And so there was an absolutely extraordinary atmosphere at the Pasteur Institute, and I arrived there as a doctor. Well, it wasn't always very comfortable because at the time, and unfortunately it still exists a bit, a kind of condescension, not to call it contempt, from the medical community towards researchers, because for the top doctors, researchers were seen as a bit of a joke, And then there were researchers who thought doctors were useless at science. Well, I'm exaggerating a bit, but there was some truth to it. So from the very beginning, I found myself a bit behind, caught between two worlds, so to speak. But it wasn't that uncomfortable because it was actually pretty funny to watch these two groups judge each other. And so I got into that. And the path, well, maybe that's what decided my career. And let's say the recognition I received in science was about saying you really have to build a bridge between those two worlds. On one side, there's very fundamental biology. So at the time, genetics of E. coli, and what can we do with that in medicine? And there was starting to be the whole problem. So at the beginning of the 1970s of antibiotic resistance, because antibiotics had existed since the 1940s and 50s, miracle. And then disaster, we started to see very quickly with the graphs. The more antibiotics we produced and used, the more resistance increased. And so there started to be a real approach to genetics, meaning what's behind it. Why do bacteria become resistant to antibiotics? Why are there no exceptions? Why will every new antibiotic be subject to resistance mechanisms in bacteria? And for that, you had to do genetics. So I started with that. And then very quickly, I found it a bit routine. One antibiotic, one resistance, one antibiotic, one resistance. I thought to myself, why not also try to understand what is called pathogenicity? Why is a microbe pathogenic? Why is an Escherichia coli harmless, circulating in the digestive tract without any problem? And why are other strains, the Escherichia coli, pathogenic and sometimes cause serious diseases? And the microbe I started working on is called Shigella, the Shigella, which is responsible for bacillary dysentery. It's a disease that is quite serious in children, especially in the most underprivileged countries. Why is that? Because Shigella basically is part of the Escherichia coli species. We're in the same microbial world, taxonomically speaking, as we say in microbiology and other disciplines. So the idea was to ask ourselves basically what's going on, what's the difference? And that's where my work really began. Identifying the genes that allowed one microbe compared to another to be pathogenic in humans. I discovered a plasmid, an extra chromosomal element. These are genetic elements that replicate in the bacterium independently of the chromosome and often carry genes that give bacteria their functions. To help them adapt to their environment, since they haven't integrated it into their chromosome yet, they capture it from a nearby element. And that's when I started to identify this concept of virulence genes that were acquired from the environment, from other microorganisms. It's probably exciting too. And that the bacterium had finally adopted and made functional in order to use them. For its pathogenic needs, because a bacterium is pathogenic not for the pleasure of hurting children or making people sick. In general, it is pathogenic because it's Darwinian, it needs to survive, and in order to survive, if it didn't have these pathogenic elements, it would be at a selective disadvantage compared to other microbes. And so this kind of massive pressure, which maintained and probably helped build a sort of mosaic of pathogenicity genes, basically programmed the disease as we observe it. So that fascinated me. I went to the United States for 2 years. There were very few groups at that time, we're talking about the late 1970s, that were really interested in pathogenicity. And that allowed me to quickly get in touch and establish friendly relationships with the few scientists in the United States who were already working in that field. And it's quite fascinating because it really created a kind of camaraderie. We supported each other over the years. And when I returned to the Pasteur Institute in 1981 in the last century, as I tell my grandchildren, I already had this program firmly in my mind, and I continued to care for patients until 2000. At that time, there was a hospital at the Pasteur Institute, which was an infectious diseases hospital. we found ourselves caught from the early 1980s in the whirlwind of AIDS. So I took care of the clinical side, setting things up, welcoming the patients, because I don't know if you might have known that period. It was a bit early for me. It was a bit early. But the period of 82 to 83, when AIDS really started to develop in France, and in 95, 96, 97, when the treatments arrived, that's about 10 years later, even 15 years, it was war. It was really war. It was terrible. You were talking earlier about infectious diseases in immunocompromised patients. It was absolutely disastrous. Lung infections, viral infections, cytomegalovirus. It was a nightmare for these patients. And so we tried at Pasteur Hospital, as in other centers, of course, to provide the best possible care. There was the discovery of the virus at the Pasteur Institute. It was a period that was both intellectually exciting, but on a human level, it was really very, very complicated. So I found myself caught up in it. I was talking about schizophrenia. As I said earlier, it was a bit difficult to take care of my laboratory work on one hand, and then these patients for whom I had to be available all the time. Looking back, family-wise, it was a bit complicated, but that's how it was. In a way, I'm proud of it because I managed to do it, maybe like many of my colleagues who did it too, to keep that kind of kindness, openness towards patients, and maybe a more sophisticated way of working, if I can say that, in the lab. And then the work went well. We found some interesting things, really, to identify all the genes that allow a microbe to enter a cell, which is a severity factor. Of course, you can sneak in like that. Inside the cells, in this case, it was the cells of the intestinal epithelium, which is the cellular layer that protects and gives function to the intestine. So that's this kind of mechanism that allowed, we invented, so to speak, a new discipline that we called cellular microbiology, which means how microbes, bacteria, but also viruses, of course, can go, make them adhere, attached to the surface of a cell. What are the two sides, the ligands, the receptors that allow this specific interaction, and then how the cell. Finally, we call this endocytosis. It captures these microorganisms, allows them to enter Shigella. Also, it's interesting because usually when microorganisms, bacteria, are captured by cells, like by polymorphonuclear cells, for example, the cells, the phagocytes in the blood or tissues, they stay in vacuoles, in membranes inside which enzymes destroy the bacteria. Here, Shigella had the ability to destroy this membrane and move around in the cytosol, that is, in the cytoplasm itself of the cell. It could move inside the cytoplasm, and it could go from one cell to another. And all of this was related to the fact that the bacterium was able to interact with what we call the cytoskeleton, that is, all the molecules, especially actin and its regulators, which basically create the dynamics of the cell in terms of its ability to move, for example, or its ability to attach and stabilize within a tissue. All of that needs to be structured, and this actin in particular, and myosin, which enables the movement of these actin filaments, it's extraordinary. There were discoveries almost every week. In English, we say harness. It was really domesticated by the microbe for its own interest to invade a cell and to invade adjacent cells. So today, it seems almost natural, but at the time it wasn't obvious. And that also led us quite quickly to realize over the years, when Metchnikoff discovered cellular immunity, that is phagocytosis by macrophages, the capture of microbes by macrophages, at the end of the 19th century, When he came to the Pasteur Institute, there were a number of things we didn't understand, and immunity was just beginning to emerge. We were just beginning to learn about antibodies, so we were also starting to learn about these phagocytic cells, and the immune system was just starting to be conceptualized. In terms of really precise specificities, for example, an antibody that neutralized a microbial toxin, that was relatively clear. But what we didn't understand was how the human or animal host knew or perceived that it was being attacked by a microbe. And that the immune system, as we were beginning to conceptualize it, what we call adaptive immunity, didn't really provide an answer. It had important, significant effects on survival, important effects on what it did. It's a relatively slow process, which takes hours or days for a lymphocyte to be able to produce antibodies, for example, that are specific to a given antigen. It still takes time, and we don't have that time. As an infected individual, you need to recognize it right away. And so there was all this research that made it possible to recognize and identify what we call toll-like receptors, which are molecules that detect microbial patterns. That made it possible to sense the presence of pathogenic bacteria, not based on the billions d'antigens that a microbe can present, but rather on a few that were intrinsic elements of its structure, its wall, its surface. So that was the discovery of tolls. This was actually rewarded with the Nobel Prize for Jules Hofmann and Horace Beutler in 2008.
Let me pause for a moment to make sure we understand. If I use an image, it's like antibodies are able to recognize an infected cell just by touching it, by seeing what it looks like on the surface?
Antibodies. They will recognize an element on the bacterial surface or an element of a toxin, a product secreted by the bacteria, and neutralize it. So that's specific immunity. Here we're talking about another area that's related, of course, but it's what we call innate immunity, not adaptive immunity, meaning this immediate recognition of the presence of a microbe on the surface of a cell, on the surface of a tissue or within a tissue. And this recognition, that was all the work on bacteria, on the toll-like receptors I mentioned, which are not systems that work like antibodies, meaning there's no adaptation to a given antigen. There is an immediate innate recognition of patterns on bacteria or on the virus potentially. It's a real change in the way we see the immune system in complete complementarity, because this innate immunity allows us to recognize the presence of a microbe. It allows the triggering of inflammation mechanisms, which are the first defense mechanisms against an invading microorganism. And so it sets the adaptive immune system on the path to its own response, because this innate response leads to the secretion of molecules that stimulate the functioning of the adaptive immune system. All right, so we're dealing with a system of two interacting boxes. And so this innate immunity, which was suspected but not very well understood, became clearer at that point.
And Shigella,
in particular, being intracellular. Now it has shown us that there are mechanisms for sensing bacteria and then viruses, not only on the surface of cells to try to explore the environment, but also inside the cells. This allowed the host, in addition to being warned that there were bacteria around, to know that things were going wrong because the bacteria had entered the tissues, and that's not really where they belong. There we discovered a new family of molecules called nod molecules. I won't go into the details, but they allow the cell to sense from the inside the presence of a pathogenic microorganism, a bacterium in this case. But there are whole families of equivalent receptors that can detect viruses, that can detect fungi or any other pathogens you can imagine.
So, as a side note, what's the point for the cell to know that it's being contaminated by a pathogen? How does it react?
It's interesting to look at it from that angle. If you will, it's essential. We're surrounded by microbes. We live among microbes. We have microbes inside our bodies. We have 100 trillion of them in our intestines, yet we're not really harmed by them. So there's some kind of deal that's been made that means the host and the microbe have signed a contract for mutual living, where basically it's minimal aggression, minimal adapted response, and we don't even notice it, right? Otherwise, it would be terrible. Life would be impossible. So the question is, when do microbes cross the line? And that's where this new element comes in, which is pathogenicity. That means the microbe will no longer respect this deal with the host. It will attack the host. A current one, but well, there you go.
And then
you have to respond, because otherwise the host will be overwhelmed by this infectious process
and
will be destroyed, both by the microbe, which has its own intrinsic destructive mechanisms, enzymes that break down important proteins in the tissue, in the cell, and then by the massive inflammatory reaction. Often, what kills us in infectious diseases or makes us very sick in infectious diseases is, of course, the microbe that triggered the process. But it's sometimes our own excessive response to that microbe. So that's an essential element that defines pathogenicity, both the aggression of the microbe and the response, sometimes excessive but essential, of the host. Because if you let the microbe keep doing its little job of invading everywhere, you're dead. So that's where the whole debate lies, and that's where you basically ultimately define the margin that exists between what we call a commensal, which often isn't very different from a pathogen as seen by the host, and then the pathogen which has these aggressive mechanisms that the commensal doesn't have.
All right, so in fact, there is, in quotes, a sort of tacit agreement between the bacteria and us who coexist.
And actually,
the immune defense we have is just a kind of safeguard that helps maintain this balance.
It's a safeguard that works all the time, because we can't imagine that there aren't small border violations happening every day, because there are a few too many microbes accumulating in one place. And then there are microbes whose strategy isn't clear. They're kind of in between the two. And if the immune system works well, you don't hear about them. If the immune system doesn't work so well, they can become pathogenic, opportunists in particular. So there's a constant interplay that we don't realize on a daily basis, but it's essential. And basically, the presence of these commensal microbes is important because it keeps the immune system alert, regulated within limits that are compatible with life and daily comfort. But the advantage of this system, which is constantly on alert, is that it's not neutral. It's not microbes on one side and us on the other without communication. There is a daily dialogue, but it's controlled. If there is a breakdown in this system, of course, we get the inflammatory response, particularly the influx of defense cells and so on. And there you have it, it's war. So these are systems. Innate immunity is important because basically when you come into accidental contact with a pathogenic microbe, you don't have to reinvent the wheel every time or go through absolutely all the defense steps again. The system is ready to work, and it works very, very quickly. And that's what's pretty amazing, and that's what fascinated me very quickly. It's about being on both sides. So people would tell me, you think like a bacterium, but at some point you have to put yourself in the shoes of the bacterium. I like microbes. Besides, less than 1% of microbes are pathogenic. All the rest, we can also talk about the biosphere. 16% of the planet in terms of biomass is microbes, so we can't imagine that they're all pathogens. On the contrary. But so there you go, it was becoming very interesting. And actually the next part of my career is the European Research Council came along, the ERC, and we started to get large European funding for projects that were a bit at the forefront of microbe-host relationships. And that's when we really started to take an interest in commensals, to ask basically what makes a bacterium pathogenic compared to a non-pathogenic bacterium. Let's also take an interest in what these commensal microorganisms do, especially when the human genome was sequenced in 2004. We immediately took a position by saying, like, wait, the sequencing of the human genome is great. It's the eukaryotic genome. It's our cells. It's our hands. But we should also perhaps consider that we are actually a metagenome, meaning that we are both a human and eukaryotic genome. And then we are also a microbial genome, a series of microbial genomes, intestinal microbiomes, skin microbiomes, respiratory, nasopharyngeal, and major vaginal microbiomes. In women's health, the vaginal microbiome. So precisely this relationship, I would say, may be less combative and more subtle between the microbiome, the commensals, and the host. What is its true nature, and what exactly are these mechanisms? Both controlled aggressions and measured responses from the host, how these microbes potentially impact beyond the immune system development, stem cells, and tissue repair. How ultimately do these microorganisms, which still cause confusion, work? They stimulate the immune system, and especially innate immunity. Inflammation. One of the effects of inflammation is also DNA breaks in cells that are very sensitive, for example, to oxidative stress. And if these repairs are not done properly, a process of cancerization can occur, especially in stem cells. Because the stem cell is there, it is immortal. In quotes, the constant battering of these small local inflammatory reactions related to the conflict, a conflict that can be relatively minor, but still. And from there we got to cancer, and so basically a shift in pathogenicity seen as acute infectious disease towards a shift to chronicity. Pathogenic processes linked to the host interface, and in particular to chronic inflammatory diseases and cancer. And this is a real current field that is developing, which is the understanding of the mechanisms of commensalism. And because beyond the very positive view we have of commensalism, these gut microbes, for example, which make up the largest mass, play a role in nutrition, metabolism, and development. There's even the famous gut-brain axis, where gut microbes also influence behavior. And the mother's microbiome even influences embryonic and fetal development. So there are a whole lot of factors that make us a meta-organism. Once again, as we said before, with our animal cells, eukaryotes, and then our microbial cells. But so there's this whole new world opening up with minor disruptions. And what's interesting is that these disruptions happen at the interface between the commensals. As far as we're concerned, they are largely favored by the modern lifestyle. Okay, let's say junk food, to simplify things a bit, is a destabilizing factor for the gut microbiota in that it brings excessive amounts of fats, proteins, and sugars that humans have never been exposed to before. And so in terms of coevolution, because all of this is a matter of evolution, of course, we've reached a balance over the millennia with a pattern of gut microbiota. So it's always the same microbes, but it's the same functions that these microbes provide. And all of a sudden we destabilize the system by flooding it with an excess of substances, even completely foreign substances, emulsifiers, things that will completely disrupt it. At the same time, the composition of this microbiota is reduced and human microbial biodiversity is gradually decreasing. And then it leaves empty spaces for those well-known microbes called pathobionts, which will lead to what we now call low-grade chronic inflammation, which is a kind of situation where the innate system is raised to a higher level, even if it's not noticeable. And behind all that, we have the effects of chronic systemic inflammation, meaning obesity, diabetes, certain cancers, autoimmune diseases, allergic diseases, and asthma in particular. All of this is connected, at least in part, not exclusively, but clearly encouraged by the modern lifestyle and by the disruption at the interface between our microbial world and ourselves. As a eukaryotic organism, these are truly fascinating aspects of microbiology that take microbiology beyond the strict field of infectious diseases, which people sometimes find a bit boring because it's a bit repetitive. Even though COVID has shown us that nature is full of originality in its ability to bother the human species, so microbiology is becoming, I think that if Pasteur were here today, he would rush to address these issues. Infectious diseases of the microbiome and the interaction of chronic diseases. There you have it. That's a bit like our new world, microbes. And the mistake would be to forget about infectious diseases because they make sure to remind us every day. Well, not every day. Fortunately, things are unfortunately not settled.
But if I understand correctly, then all microbes, the entire microbiome, have quite a significant impact on health in general and on many of the ailments that people have right now.
That is to say, we have to be careful in the way we present it because we shouldn't present it in a negative way. To the extent that, once again, we are a meta-organism, we are both eukaryote and prokaryote, and we live thanks to that. And this was seen quite quickly in the 1950s and 60s when we were able to start developing these methods in a really rigorous and almost industrial way. What we call axony, that is raising animals in controlled microbial conditions, including what we call germ-free animals, which means basically creating a fetus and then raising it in those conditions. In the uterus, it is aseptic, it has no microbes, and so if we manage to have a mouse give birth in a completely aseptic environment, we will have produced offspring that are germ-free. And we quickly notice that these offspring are handicapped at birth. They have completely unusual behaviors, such as loss of bearings, loss of risk perception, and a risk of memory loss. They constantly re-explore their environment, so we can clearly see that there is brain immaturity when we look at their immune system. It's absent, meaning there are no lymph nodes. For example, loss of risk, loss, not anywhere else either. The spleen is atrophic, so they don't have development of their immune system. They don't encounter microbes. Their metabolism is insufficiently organized. They are hypertrophic. They grow very little. They are smaller than their peers who have a microbial flora. So we are able to identify by comparing animals often referred to by the English term germ-free to animals that have a normal microbial flora. And in this delta, in this systemic difference, because once again it affects the nervous system, the immune system, immunity, the endocrine system, and whatever else you want, it's systemic. There you have the delta, the difference that microbes bring us. So we are a microbial being just as much as we are a eukaryotic being, and that's what's fascinating. So that's the positive side, and that's the everyday side. Now that encourages us to be extremely careful, precisely, and to take care of this microbial world. If we attack it, it's not that it defends itself, it's simply that it deteriorates. And so as it deteriorates, we almost go back, I would say, to the germ-free mouse. That is, we lose the functions that these microbes provide us, which others bring us, but this time they are negative. In the sense that it's pathogenic, there are microbes that come to fill the void. So we see these so-called pathobionts appear and develop. That is to say, these microbes that are halfway between commensalism and pathogenicity. And that will program the emergence of a tumor cell, in particular, probably a tumor stem cell. So there you have it. This is the new microbiology, and I'm passionate about it. As a retiree, I have a bit of trouble going further, and I supervise. We try to keep working and to help promote this new microbiology, which is essential. Basically, we've almost controlled infectious diseases, especially with antibiotics and vaccines. And we find ourselves with another area of the human microbe interface, which is the breakdown of the human commensal relationship.
I think it's interesting to talk about because right now I see a lot of people rejecting it. So all of medicine, medications, is the breakdown of the relationship. Since we need microbes for our bodies to function well, reducing the amount of medication would allow us to have more microbial input. Does that make sense, or on the contrary, is it counterproductive? How can we help maintain our flora, meaning intestinal flora, microbial flora, in the broad sense?
And no. Sometimes there's a bit of ideology as well, because it's those who refuse the advances of medicine, like vaccines, for example. And these attitudes, often borderline or even completely conspiratorial, these attitudes of alternative truth and so on. It's true that for us scientists, at least, it's kind of the poison of the century. And we need to try to restore a bit of an objective view of all this. Nevertheless, it's clear that the modern world and these chronic diseases we've been talking about are also called postmodern diseases. It's linked to the progress that man has introduced. No one says that antibiotics don't alter the gut flora, so fortunately it can rebuild itself quite easily, but if you use them too regularly, we end up contributing to this ecological alteration of the internal microbial environment that characterizes us and on which we've built ourselves. There are other medications as well that have antimicrobial effects, side effects, but on the microbes. The question isn't to immediately reject the progress that antibiotics have brought. First, it's about using them wisely, only when it's absolutely necessary. And by doing that, we will have already contributed greatly to improving the situation. It's also about avoiding their use. Absurd. Antibiotics, outside of clearly defined infectious diseases in humans and animals, meaning any other use of antibiotics for comfort in humans, even in animals, and then absurd use. Growth, for example, of livestock in intensive farming that pollutes the environment. It damages not only our internal microbial environment, but also the microbial environment of the land and water. There are now antibiotics in river waters in small doses. They still promote the persistence of resistance in a number of bacteria. I think we need to stay alert. We need to identify, in any case, the pitfalls and negative elements that progress can bring. Integrating, ultimately inserting into our daily lives. It's the same thing for certain foods. All processed and industrially made foods and so on have these negative effects, especially on the gut flora. And clearly, a return to more traditional diets with more fiber, less rich in protein and in fats, especially fats that we don't usually have in our diet. So, all of this requires us to adapt, but it doesn't mean we should reject progress. Progress is here, and by definition, we can't stop it, but we need to understand it and stay alert. We need to teach, educate, and explain to children that dietary rules are what they are, and there are reasons why they need to be followed. Not just because we should be nostalgic about traditional food. No, there's a reason behind it, and I think that's important. It's important to make people understand this concept of coevolution, truly our human-microbe interface. It was born from thousands and thousands of years of mutual familiarity. And if all of a sudden the rules of the game are changed by the environment, the new environment we're exposed to, there will be consequences.
So, regarding the rational use of antibiotics, we're often told that we shouldn't necessarily take antibiotics if we have a viral illness. Can we explain again the difference between a bacterium and a virus? And maybe understand how an antibiotic works on bacteria to stay on the bacterial side?
So an antibiotic is a molecule that targets a key mechanism for a microbe's survival or its ability to multiply. In the end, it's basically the same since it'll disappear anyway. So it can be something that blocks the synthesis of the microbe's cell wall or that blocks its metabolism. For example, it will attach to the ribosome and block the synthesis of the microbe's proteins. All kinds of possible targets for antibiotics. These molecules don't work on viruses because viruses have a completely different behavior, and on top of that, they are totally dependent on a cell. Viruses wouldn't exist if there weren't human, animal, or plant cells, by the way, since there are plant viruses too. To multiply, they lack all the machinery needed for reproduction, so they can only do it through a host cell. That's why antivirals are completely different molecules from antibiotics. And they will block, if possible, in a very specific way, this signaling area between the cell and the virus, which allows the virus to replicate inside the cell. So that being said, there is antibiotic resistance in bacteria, and there is also antiviral resistance in viruses. But it's less of a problem because it's less common and we use antivirals less often. Overall, they are more targeted at certain very specific diseases than antibiotics are. So yes, if you use antibiotics for a viral illness, it's useless. Except in special situations where there is a secondary infection by a bacteria, for example, measles and pneumococcal pneumonia, because measles weakens the lungs and pneumococcus will develop, so you will need to give antibiotics. But in everyday medical practice, just because you have a sore throat doesn't mean you need to take antibiotics. It can be, and usually is, a purely viral infection, a viral nasopharyngitis. And antibiotics in this case, first, they won't do anything to change the course of the infection, and second, on the contrary, they'll unfortunately have negative effects on the microbial flora, which I mentioned and we've discussed the reality of, but also the possible price to pay. The problem is, first of all, educating medical staff to know that it's not good to take antibiotics when they're not needed. It's also about distinguishing between viral and bacterial infections. When we're faced with this child, baby, or any other person who says, "My throat hurts," and that's not always easy. Science helps because now we have, for example, one of the problems if we look at the throat. It wasn't necessarily an insignificant choice, but on top of that, it's so common, it's every day. There are two possibilities, roughly speaking, to keep it simple. Either it's a viral infection or it's an infection with a streptococcus, what we call beta hemolytic streptococcus, a streptococcus that causes a sore throat. And now we have what are called diagnostic tests. At the patient's bedside or with a test strip, we can tell if it's a streptococcus or if it's probably a virus. Based on that, the doctor can decide whether to give antibiotics. So we shouldn't have the attitude of saying you don't understand anything, you're using antibiotics inappropriately. Making that decision at the patient's bedside isn't always easy in the moment. Scientific discoveries, knowledge of biological models, immunodiagnostics, strip systems or PCR, which can now be done in a relatively simple way, with tools that are almost usable at the patient's bedside. All of this helps to inform and guide decision-making, especially when it comes to taking antibiotics.
Here, we've talked about antibiotics, but sometimes antibiotics aren't enough, and that's when vaccines can help solve certain problems. How does a vaccine actually work?
Oh, well, basically, if we go back to the Pasteurian method, a vaccine is simply about injecting a person with a microbe or a piece of a microbe, to put it simply, which will allow them to produce antibodies without actually causing the disease. There's also the cellular response, which we can quickly revisit, and these two responses are complementary, which will allow the body to eliminate the real microbe when it comes into contact with it, because the body already recognizes it. It already has the cells that are able to very quickly produce the specific antibodies for that microbe, and cells that are called T cells, to recognize cells infected by that microbe and destroy them. Anyway, that's what it's for. The vaccine is meant to anticipate a future infection by the real microbe by educating the individual's immune system to respond to it. So we don't have to respond to all the antigens or molecules of the microbe in question. We respond to key molecules. For example, against pneumococcus or Haemophilus influenzae, which cause very severe pneumonia and very severe laryngitis in infants. In fact, in this vaccine, we include an element of the microbe, which is its capsule. These are complex sugars that cover the microorganism and protect it against innate defenses. We talked about this earlier. These capsules make the microbes slip past the polymorphonuclear cells, the cells that would like to catch and phagocytize them. So what the vaccine will do with this capsule is that we will make antibodies against this capsule. And these antibodies will be able to attach to the microbe when the real microbe arrives. And thanks to these antibodies, the phagocytic cells will recognize it. And this time they will be able to engulf them and destroy them. That's what the vaccine is for. It allows the host to anticipate a future infection by using an element of the microbe that doesn't itself cause disease. So vaccines can be made against bacteria, they can be made against viruses, and they can be made against parasites. The system, the principle is the same. Some vaccines are not always very well tolerated. That's what causes there to sometimes be rejection or concerns, especially from parents when it comes to vaccinating children. This is because the molecules we inject are not always as effective at stimulating the immune system as the real microbe causing the infection would be. So we have to add what are called adjuvants, which are systems, molecules that can be aluminum or specific lipids. These will stimulate the innate immune system we talked about at the beginning, which will allow a more effective immune response to the vaccine antigen that was injected into the person. And this reaction can cause local swelling and fever. Well, let's say that's, in quotes, at a cost to avoid having these infections that are dramatic. And the idea, perhaps, of many people who are against vaccines is, but why vaccinate against diseases we don't see? Honestly, it's absurd because we don't see them because people are vaccinated. It's not that we don't see them because, as a matter of fact, we had the case of Venezuela recently. It's a country that collapsed economically in just a few years. The doctors left and vaccination campaigns were stopped within a few months, years. We saw diseases reappear in Venezuela, which was a country with Western-level sanitary conditions, with vaccinated people and doctors. We saw diphtheria reappear, we saw measles reappear. On a vu réapparaître le paludisme. It's not a problem with vaccines, but because the malaria eradication campaigns were stopped. So infectious diseases are still here. The microbes are still here. They haven't disappeared. They're simply kept in check thanks to vaccines. In any case, when there is a vaccine, by regularly vaccinating children in particular, we establish herd immunity that protects our societies. In addition to hygiene and, of course, a number of other behaviors that are essential.
In fact, it's a bit of a vicious circle, because the more progress we make in terms of vaccination, the less we get sick and the less we tend to realize the importance of vaccination.
One example is polio. For me, my generation, my parents were terrified of poliomyelitis every year. In the summer, because it was a summer disease, it was kind of like the summer flu, poliomyelitis would arrive, and there were tens of thousands in France, tens of thousands of cases. Fortunately, most of the time, there were no major consequences. Once again, it was kind of like the summer flu, but very often there were first complications, paralysis, atrophies for the rest of one's life, and then deaths. And it was terrifying. I remember my mother, her terror. In the summer, it was about catching polio. And when the polio vaccine was used, available in France, it started in 1957. People rushed, I still remember it, to get their children vaccinated because it was unbearable. And polio, with a lot of difficulties in the global south, because logistically it was quite hard to achieve. Polio has almost disappeared, or even disappeared, except in countries that refuse polio vaccination. In short, you can always find exceptions, but there you have it. Polio has disappeared, smallpox has disappeared, measles has disappeared, well, almost. But you have to vaccinate the population at a high level, otherwise you see cases reappear. We're seeing them again in France right now, and we'll see them again in the United States, since the brains of the Trump administration have decided that they shouldn't vaccinate against measles anymore. Well, there you go. They're going to rediscover the delights of little children getting severe pneumonia and encephalitis. And when stupidity reaches that level, for us doctors and scientists, it's a great sadness.
I feel like there are a lot more infectious diseases in tropical countries. Is it for biological reasons, meaning that microbes develop better in those regions, or is it more because of vaccination reasons? Vaccines are more complicated.
Well, yes and no, that's the problem. First, there's an epidemiological issue. Diseases exist in southern countries that aren't found in the north, purely due to climate, like malaria. Which actually used to exist in our regions, but is greatly facilitated by the fact that the climatic conditions largely help the development of mosquitoes that carry the... There are reasons that are purely and which may actually be disrupted by climate change and the warming of global temperatures, because these populations of vectors, especially mosquitoes and arthropods, will spread more into northern regions where the temperature is rising. So that's the first reason. The second reason, unfortunately, it's often the level of sanitation that is insufficient, especially in the most vulnerable countries, those with the lowest GDP. And among the elements of this health approach to infectious diseases, there is vaccination. So in recent decades, there has been enormous progress in the development of what is called the expanded vaccination program, which is a matter of morality. I mean, why shouldn't the little children of the South have the same treatment and the same vaccination as the little children of the North? And with international aid, many countries have been able to do so. Besides, the African continent is now trying to achieve its sovereignty when it comes to vaccines. And there are development programs that rely on countries that are a bit more technologically advanced, like South Africa, for example, or Egypt. So that Africans, pas totalement dépendants, can Western pharmaceutical industry. And that's encouraged because there's no reason. There has to be sharing, including knowledge, and not just the use of vaccines. So the situation has improved considerably in Africa, but it is extremely fragile for some countries. And once again, the reduction of aid to developing countries, as we say, due to some recent North American decisions, makes us a bit worried. Regarding the sustainability and durability of these vaccination efforts, we must be uncompromising about this. Children in the global south must be vaccinated just as children in the global north are.
I heard that the COVID vaccine is different from traditional vaccines. There are different vaccination technologies.
Of course, there are two main types of vaccines, what we call particulate vaccines and live attenuated vaccines. For example, the measles vaccine is a strain of the measles virus that has been passaged, and repassaged many times, and its virulence is attenuated. It's like rabies, like Pasteur's historic vaccine, so there are quite a few of them, especially the one against measles as far as Western countries are concerned. For countries in the south, for example, the yellow fever vaccine is also a vaccine made from a live attenuated virus. And for polio, there are both. There is a killed vaccine and a live attenuated vaccine. So that's one possibility. The other possibility is indeed to have killed vaccines. It can be either killed bacteria or killed viruses. The polio vaccine, for example, is a killed virus. There's nothing more to it. We administer all that and it works well. And then some are not well tolerated. For example, the killed vaccine against whooping cough was not well tolerated. So we made it a bit more sophisticated. We took specific antigens, what we call subunit vaccines, and we vaccinate like that. But it still remains in the realm of killed vaccines. We don't inject live microorganisms. So those have been the two main approaches to vaccination over the past decades. And then there's this new approach with the messenger RNA vaccine, which is a little bit different. Nevertheless, what we inject is still a substance. It's dead, it's not a living cell. But it has an intrinsic biological action that allows the individual, since it's messenger RNA, to produce the protein that will vaccinate the person against the viral infection. This was the case for COVID. So it's a new approach that comes from the initial stages of developing gene therapy methods, but it finds its place in vaccination. It's not clear yet because other trials on other viruses are not as successful as what we observed with COVID. So we're still in a waiting situation regarding the real place that messenger RNA vaccination will take. That being said, as usual, the more options we have, the more possibilities there are, the better chance we'll have to be effective in finding the right solution. In a situation like a pandemic, for example.
In the coming years, what kind of discoveries are expected in the field of microbiology?
As for ongoing research, I would say that if we're talking about knowledge, it's impossible to give you a summary because we need to understand everything better. If we focus on the field of diagnostics, I think there is real fundamental and applied work being done on developing ultra-rapid diagnostic tools right at the patient's bedside. For infectious diseases, I believe that would considerably change the speed of development. Well, not the development, but the implementation of a treatment for a patient, especially in the global south, in countries where laboratory facilities are not always available on site. So anything related to rapid diagnostics is definitely an area where there is progress to be made and science to be applied, because advanced technologies need to be adapted for on-site diagnostics. And then there is a need for progress in the use of new antibiotics. That's clear. With antibiotic resistance, we need to find new antibiotics, which is very difficult. There are also opportunities in the use of bacteriophages, which are viruses that infect bacteria and can be a substitute, but it's not obvious that this will replace antibiotics. Small molecules can go everywhere in the body and kill microbes wherever they are, whereas bacteriophages will have a bit more difficulty, except if they are applied to surfaces. To reach every corner of the body, there are a lot of new approaches to improve and protect against infections, as well as antivirals. There are still whole areas of viral infections that do not benefit from highly effective antivirals. We now have very effective ones against hepatitis, which is great, but for the vast majority of viruses, we still don't have drugs that are specific enough. Experience has shown that there is very rarely the possibility to use an antiviral for one virus against another virus. It's really necessary. There are such specificities in the virus and human or animal cell pairing that we need very specific molecules. So there is immunity that we call for help. There are monoclonal antibodies. We can temporarily replace the immune system in immunocompromised people, for example, by giving them antibodies that will neutralize a virus. But, well, all of this will need to be organized a bit. There are needs in diagnosis, in therapeutics, and then in vaccination. There still isn't a really good vaccine against malaria, and there still isn't a really good vaccine against dengue, while this viral disease transmitted by mosquitoes is even starting to invade our regions.
But then why is it so difficult to create a vaccine? If in fact you just have to quote unquote send some kind of pieces of the disease?
Isn't that enough? It doesn't always work. There's still a black box. That's probably also where medical and medical scientific progress lies. Vaccination against dengue, for example, I mention it because it's becoming a real concern. There are starting to be cases of dengue in mainland France because of the tiger mosquito. We still haven't managed to make a good vaccine. The inactivated vaccine doesn't work. The vaccine based on surface proteins doesn't work well. And the mRNA vaccine doesn't seem to work well either. There's also a problem, which is that if the immunity that's induced is insufficient, it causes in some people an effect that actually facilitates infection when they are infected by the virus itself. We know this from people who have had mild forms, not vaccinated people, but those with mild forms of dengue. They can develop very severe forms with what we call hemorrhagic dengue, massive bleeding. So it's complicated, and that's why it's important to encourage science. It doesn't just fall into our laps, ready-made. And the main principles of vaccination are not as simple as we've described here.
How long does it usually take to develop a vaccine?
That's a good question. On average, it takes 10 years, 12 years. What was phenomenal with COVID is that in 6 months, it was solved thanks to mRNA and also thanks to the knowledge of the antigen. We already knew it before the pandemic started. We already knew the main antigen, this spike protein, the protective antigen. So there wasn't the time needed to discover what had to be put in the vaccine. We already knew that. And since messenger RNA allows you to do it directly, it worked very well. But we shouldn't think that all vaccines will be as easy, well, easy in quotes, or as quick to develop, because it was an exceptional adventure which also required a lot of money.
For those who would like to go a little further in this field and better understand the world of microbes and viruses, do you have any books to recommend? Whether it's books you've written or books that might be interesting.
I'm almost embarrassed to recommend my own books, but it's true that I try to write about vaccines. And in Microbes Without Borders, a real overview of infectious diseases and the situation in terms of epidemiology of antibiotics. For example, antibiotic resistance. How do we overcome antibiotic resistance? Well, the issue of viral infections. So I tried to explain to really help people understand the factors behind the emergence of infectious diseases in general, and also to identify the dead ends we're currently facing. Very often, due to a lack of support and recognition of how important this is by our governments, for example, so all of that is present. There's a nice book that was recently published by one of my colleagues from the Pasteur Institute about bacteriophages, a good overview of bacteriophages and their potential use in diseases. Bacterial diseases that are resistant to antibiotics. Then, well, I think it's a bit difficult to name them. There are quite a few books on infectious diseases. Maybe not enough, to be honest, because the field really needs to be updated and we need to try to present it. There are also books on the microbiome. There was a book by Patrice Debray a few years ago, an interesting one about the microbiome. But the microbiome is evolving so quickly now that things need to be readjusted and updated. Well, if you look carefully, you can find some, but it's not always easy. When you search for information about vaccines, it's much easier to find anti-vaccine books than quote-unquote ones. Even though I don't like the term pro-vaccine, I read a series of them when I wrote my book about vaccines in 2016, 2017. It was appalling for a scientist.
I imagine. Okay, so we'll put all these books in the description. We'll also add the link to the Pasteur Institute website. Yes.
The Pasteur Institute website has plenty of information.
Thank you very much, in any case, for this interview, which was extremely interesting.
Thank you.